Hello and welcome to Sa Biology class. Friends, today we shall discuss about different types of electrophoresis. Before that, I request all of you, my friends, please subscribe my YouTube channel. Also watch, like and share my videos with your friends. Electrophoresis is a technique that is used to separate particles, molecules and ions on the basis of their size, shape and net charge. It can be divided into two groups. One is free electrophoresis and the other is zone electrophoresis. Free electrophoresis doesn't employ any stabilizing media. It is of two types. One is microelectrophoresis and the other is moving boundary electrophoresis. Microelectrophoresis is used to observe the movement of small particles such as red blood cells, neutrophils, bacteria and others in an electric field with a microscope. The second type of free electrophoresis is moving boundary electrophoresis. Like microelectrophoresis, in this technique also electrophoresis takes place in the free solution. It is the prototype of all modern methods of electrophoresis and was first developed by Tiselius of Sweden in 1930s. This figure illustrates the separation of three different proteins X, Y and Z by moving boundary electrophoresis. The electrophoretic mobility of X is greater than that of Y and that of Y is greater than that of Z. In this technique, a suitable amount of pure buffer solution is first taken in the U-shaped observation cell. This is followed by the addition of the sample solution, after which again a small amount of pure buffer is added. The pH of the buffer solution is adjusted so that all three proteins will get net negative charge. These are two oppositely charged electrodes that are immersed in the observation cell. After electric supply, proteins X, Y and Z will migrate to the anode at a different rate depending upon their electrophoretic mobility. Because the electrophoretic mobility of X is greater than that of Y and Z, it will move fast along the electrophoretic cell, thereby forming the ascending boundary. Protein Z, whose electrophoretic mobility is less than both X and Y, will remain at last and it will form the descending boundary. By this technique, only proteins X and Z are separated. The other protein Y, which has an intermediate electrophoretic mobility, cannot be separated. In this region, you can see that there is some overlapping between X, Y and Z. During electrophoresis, there is a sharp change in refractive index at these boundaries. The refractive index, which changes along the electrophoretic cell when measured by appropriate optical devices such as Clarion optics or Rayleigh interference optics, yield patterns that indicate the direction and the relative rate of migration of various components present in the sample. For many years, moving boundary electrophoresis was very popular for quantitative analysis of complex mixture such as proteins in blood plasma. Recently, however, this technique has been superseded by different types of gel electrophoresis. The second type of electrophoresis is zone electrophoresis. It is more efficient than free electrophoresis. A few microliter of sample is sufficient for their complete separation. This technique employs different types of stabilizing media, different types of stabilizing media such as paper, cellulose acetate strip, cellulose acetate strip, and different types of gel such as starch, agarose and polyacrylamide. In this technique, after separation, the sample components are immobilized by fixation into different zones. Hence, the name is zone electrophoresis. The separated components can then be detected by staining with suitable reagent. Other methods of detection are visualization by UV light, detection by suitable enzymatic reaction, and also by autoradiography if the sample components are radiolabeled. Another advantage of zone electrophoresis is that the separated components can be eluted from the supporting medium and can then be used for further study. Based on the stabilizing media, zone electrophoresis can be divided into different types such as paper electrophoresis, cellulose acetate electrophoresis and gel electrophoresis. Friends, this is about different types of electrophoresis. For more updates, subscribe my YouTube channel, Sam Biology Class.